Good evening, as it would be if we were meeting at usual time. Tonight, I want to just go over two main topics. This is the first of two lectures, so this would be, say, the first block. And I would go over prokaryotes, and then the second block of tonight's class will be protists. Go blank, sitting here recording videos, you know what you want to say, and then all of a sudden, okay. So what I'm going to do here is go to full screen to start the lecture. Hang in with me. Bacteria are really fascinating. As a matter of fact, when I first started my undergraduate work, I was interested in bacteriogenesis, or pathogenesis of bacteria and viruses. There are more bacteria in your body than there are people in the world. Clearly, you are a habitat, and your habitat in microbiome is more diverse and different than mine. I said more diverse, but all I really meant was different than each other's, which is why we don't drink after each other all the time, right? Because the population in your mouth has adapted to you, and you will no longer get sick from them. But should they enter into mine, they have some advantage possibly that the bacteria in my mouth doesn't have. And the same in reverse. So that's why we wash hands after handling things that other people handle. You don't get sick from a lot of these bacteria that inhabit our face and our hands and everything else. But you can get sick from the bacteria of other people's hands and face and everything else. Okay. Bacteria don't only cause us to get sick, we also could not live without them. So just as I say, they hang out in our mouth, they're opportunistic and make us sick. You also could not digest and get many nutrients from your foods were it not for the bacteria in your gut. Your microflora, they call it. So this is why we introduce babies to food slowly, so that they can slowly build up that bacterial fauna, because when they're first born, they don't have any, right? So we're going to back up, and I'm going to go ahead and clear myself out, and let's get started. Okay. There we are. I don't know why I wasn't switching. Sorry. Old Kingdom. 5-kingdom system. So you read a little bit about this. We have Monera, Protista, plants, fungi, and animals. These are the ones that we use probably up to the late 1970s, and even still use them some in the 1990s. And now we use the new 3-domain system, which reflects a much greater understanding of the relationship based on molecular evidence. So there's my little sound effects. Hopefully you can see these. So we've learned about eukaryotic cells. We've seen your cheek cells under the microscope and so forth. And remember those little specks we tried to draw were bacteria, and we had purple ones and pink ones. If you remember, there were either dots or dashes. So this is going to be our focus today, are the prokaryotes. But here are the 5 kingdoms we used to think were in there. We had the prokaryotes, which are bacteria. Since then, we've found the archaebacteria. These are really crazy. And again, we'll talk about each of these in turn here. While they are prokaryotes, almost everything else resembles more of us, eukaryotes. So eukaryotes do have a nucleus, and every other kingdom, protists, plants, fungi, and animals, all are eukaryo. So these are the 3 domains: prokarya, which can be bacteria, prokarya, which can be archaebacteria, and then domain eukaryo. And all other kingdoms fall under the eukaryotes. All right, so first up, look at the size here. We're looking at 3.8 micrometers and 1.7 micrometers. From here to here, that's about the same, but when you notice this number is a lot smaller than this, these archaebacteria are really tiny, simplest. As a matter of fact, the word archae means ancient and bacteria. So we have our 6 kingdoms now. This has been split up from Monera on the previous slide to Archaea. So domain bacteria is the first of the 3 domains. And these are coming in about 3 different shapes: round, spiral, and rod shaped. That's the focus of tonight's lecture. We will hit a little bit about Archaebacteria. Archaebacteria are really extreme bacteria: halophiles, thermophiles. Thermophiles, like really hot and really cold environments. Halophiles, like really salty environments. So you find them in really weird and obscure places which you would never see unless you go deep sea diving, volcano climbing, sulfur spring spludging, and stuff like that. Those are the Archaebacteria, and these are the only 2 prokaryotes. Meaning very simple. They are pro means before, karyote referring to the body of the nucleus. There is no nucleus in bacteria of any kind, and so both of them are referred to as prokaryotes. Say that, prokaryote. All the rest are in the same domain. Now, just because you're in the same domain, doesn't mean you're so similar. Obviously, seaweed and mountain goat are quite different things, or horns. But I want you to notice something here. What it does say is that the difference between these 4 kingdoms and these here, bacteria or Archaebacteria, are as different. Meaning you are as different from Archaea as you are bacteria, and bacteria are as different from Archaebacteria as they are from other bacteria. So we're all on kind of even scale. As small and weird as Archaebacteria are, they actually share some things in common with these organisms that this kingdom bacteria does not. Anyway, just a cool point to make. Don't know if I made it. Protists. This is kind of odd. So we cannot say unicellular completely, and we can't say that bacteria and Archaebacteria are all unicellular, because it turns out protists mostly are unicellular. And what does unicellular mean? Uni means one. So one-celled organisms. We're talking very small things. You read a little bit about this if you read the microbes piece from Tuesday night's class. Here is an amoeba, and here is a paramecia, 2 very common protists. But these are these are much larger, actually, I should say, about 10 up to 100 times larger than what bacteria would be. All to say that the number of cells that make up the organism has nothing to do with which kingdom it's in. True, all of these are single-celled. All of these are single-celled of the bacteria. And eukaryotes can be multicellular, such as these fungi, plants, and animals, or they can be single-cellular, such as kingdom Protista. Kingdom Protista are very diverse. That's going to be, we'll talk more about them in the second lecture for Thursday night. All right, but let's get back to our prokaryotes. So they are in the domain bacteria and Archaebacteria, as said. Here are the 3 classification domains. Everything from here branches off is what we'll be talking about. Again, we're not going to talk too much about Archaebacteria. They're just so strange. But yes, bacteria live everywhere. All ecosystems, including your stomach, streams of water, on plants and animals, in plants and animals, in the soil microbiology. You can get very high degrees and level of professionalism just in studying the soil. They're in the depths of the oceans, hot ocean vents, that would be the Archaebacteria, in extreme cold environments, hot environments, geysers. There's no place a single cell bacterium has not been able to exploit for life. This is crazy amazing. I mean, you think about all the things you have to do to stay alive and maintain homeostasis within your body: pH, oxygen, carbon dioxide out, sugar in, waste and elimination, heart circulating these nutrients. I mean, the bacteria do all of this as one single cell. So to call them primitive might be a disservice. They're doing all the things you have to do as a single cell in very extreme environments, but obviously adapted for it. The Red Sea, the Dead Sea, there's a red algal bloom. So halophiles on the salt, on the living and on the dead. So thank you, bacteria, for cleaning the place up. And please, let's not get out of hand, as we do see oftentimes. Exactly my point, Mr. Penguin. That's what I was saying. Everywhere. So, the bacterial diversity. Rods, spheres, and spirals. Oh my! These are the only 3 shapes there are. So, bacillus are rod-shaped. Now, these are all single-celled organisms right here. They happen to link up and colonize to work together, but each one of these is a single cell, and each one of those is a single bacterium. So that's bacillus, rod-shaped. Here's streptococcus. These are round. And then you have, here's some more bacillus, or rod-shaped. And here we go, spirulina. Here's some spiral-shaped bacteria. They're all roughly the same size. They don't vary too much, but these are some really gruesome ones. Most of these are the ones you want to kind of stay away from. You might recognize salmonella, food poisoning from chickens, streptococcus, strep infection. That's not good. Staph infection, staphylococcus. Their name's so because all streps come together in 2, like a diplococcus. Staphs are clusters. So these are words. These are parts of how we classify that diversity. So, unicellular, 3 shapes: bacilli, cocci, and spirilla. Their size, about 1/10 the size of a eukaryotic cell, or 1 micron. So if that's a eukaryotic cell, that would be a prokaryotic cell. All right, their internal structure is quite simple. You can notice here in the eukaryotic cell, you got the nucleus, the mitochondrion, the ER, Golgi apparatus. What do you see inside the prokaryotic cell? No internal compartmentalization. That's right, no compartments. So they can't delegate activities to, hey, you make energy, hey, you consume the fat. They got to do one thing or another. They can make energy, ATP, or they can digest food. So they have to relegate their lively activities to one at a time. Otherwise, while you're building up molecules and you have enzymes that are digesting, they're just going to break them right back down. And all you'll be doing is spinning your wheels. So there's no compartmentalization, which means the prokaryote has to sequester its activities. So here's the basic structure. You have the circular strand of DNA. It doesn't look like a circle, but check it out. If you put a hand here and a hand here and you pulled out, this would be one big loop. You have all these ribosomes. Remember those? They make proteins. You have the cell membrane. And right outside that, you have a cell wall. Then you have the flagellum, which is different from a eukaryotic flagellum, but it moves and helps it to move. Or you may have cilia. So circular chromosome, only ribosomes, but no internal compartments. Can't fit all this inside of here, right? So this would be our chromosome of DNA, a rod-shaped. That would be a bacterial cell's DNA. Again, a loop versus a rod. We'll see more about that in a bit. So here's a prokaryote versus a eukaryote chromosome. We learned about mitosis and the double helix. Of course, it's all wound up, but then it gets wound up into these rod-shaped chromosomes. But for a prokaryote, right, we have a loop. Does that make sense? Here's the double strand. Again, it's the same, but in a eukaryote, there is an end, and there like a beginning and an end in one direction. It's a line, it's a rod, whereas in prokaryotes, it's a loop. One chromosome, that's it. Variations in cell interior. This is really crazy right here. This is a cyanobacterium, blue-green algae they call it, or it's been called. This is photosynthetic bacteria. All these membranes resemble very closely the chloroplast of plants. Isn't that cool? So they are able to photosynthesize. They have membranous sacs also that are able to intercept light, as we learned for photosynthesis. And these live, of course, in ponds and in wet environment systems. Internal membranes for photosynthesis, like a chloroplast, exactly. So there's a chloroplast, an actual chloroplast. If you look at these, yeah, it's just telling us it's made for energy, like the cristae of a mitochondrion. So the mitochondrion, chloroplast, and bacteria or cyanobacteria are about the same size as a chloroplast or mitochondria. Things to think about there. All right, so this is pretty important because when we talk about fungus and when we talk about plants, we are going to refer to a cell wall. So the prokaryotic cell wall structure is different than both the fungi and the plants. What is the prokaryotic cell wall? Very important. When you go to the doctor's office with a sore throat or an infection, you're feeling a cold, they're going to stick a swab in your mouth or in your throat or up your nose and jab it real quick. They can do this test really quick in the lab if they have their own lab, and it's called a Gram test. The Gram test is simply this. They pour a dye over it and they stain it purple. If it keeps that dye, then when we look at it under a microscope, they will look purple. But sometimes that dye rinses off and it doesn't hold on. That's because it has a different kind of cell wall. So if it retains that purple dye, we know it has this kind of a peptidoglycan cell wall. I said peptidoglycan. Glyco refers to these sugars, strands, and peptido refers to the fact that these are proteins. So you see this grid, it actually surrounds the cell membrane. So here's the plasma membrane as you've studied it in cells, and on the outside of that it has a cell wall. And that cell wall is made of peptidoglycan. It's unique to bacteria. It's a pretty thick one, and it retains that purple dye, and so when you look at it, this will tell us what kind of antibiotic we want to prescribe. Now at that point, it's up to the doctor, and when I say that, it's just whatever pharmaceutical company is offering the better trip to Hawaii, right? But being gram positive allows us to eliminate several antibiotics, and now we have a handful of about 4 to 5 antibiotics we can choose from, and we'll just give this one a shot. We always try to go with the simplest antibiotic first, something like a cyclin or a penicillin, an amoxicillin, or a tetracycline. But what happens if it doesn't? Well, it might be rinsed off, and we counter stain it, meaning we apply yet another stain that you can't see if it's gram positive because the purple is there. But if the purple washed off, you will see the pink. So pink bacteria are what we call gram negative. They also have a plasma membrane, and they have the thinner peptidoglycan cell wall, and outside of that, an outer membrane. This is crazy. So the outer membranes of lipopolysaccharides, so lipo means fatty chains, and polysaccharide is sugars. So this is a really complex cell wall, okay? And these will look pink. The way a medication like an antibiotic attacks a gram-positive bacteria is going to be different than the way it attacks a gram-negative bacteria. So bioengineers, right, biomedical scientists have to engineer these medicines to attack the bacteria in different ways. You wouldn't go up against a brick wall fortress the same way you would against a wood wall or a moat that's water or anything like that. So we have to know the kind of protection the bacteria has so we can try to counterattack it. So gram-positive, gram-negative are 2 ways we classify bacteria, particularly disease-causing bacteria, so that we can prescribe the right antibiotic, because that will kill it. Precisely. So what's another way we classify bacteria? Well, how do they get their nutrients? They're so small. Do they have a mouth? Do they have an anus? I mean, do they just imbibe it by diffusion? Yeah. So there are photoautotrophs. So if you remember, let's break down this word. Troph means to feed. Auto is self. It's a self-feeder. And if it's photo, that means it's light. They feed their cells through light. So these are going to be your photosynthetic bacteria. Okay. Anabaena, cyanobacteria. There's many different kinds of oscillatoria. This is what you find in pond scum, pond water. It doesn't even have to have scum on it. You will find this bacteria here. Very popular. Chemoautotrophs, again, self-feeders, but they feed on chemicals in the environment. So these guys oxidize inorganic compounds. This is in the soil. This is in sulfur springs. Very weird. but nonetheless, they don't need light. What they need are the chemicals of the soil. So these are deep in ground, nitrogen, sulfur, hydrogen. And they can convert these compounds into the sugars, just like photosynthetic bacteria make their own sugar, like plants do. And then they feed on that sugar in the mitochondria. Well, these can do that, except that they don't, of course, have a mitochondria. Do the same thing with chemicals. So they're called chemoautotrophs. And then lastly, the ones we're all familiar with are the heterotrophs. Heterotrophs means they feed on other things, live on plants and animal matter, dead and living, as decomposers and pathogens. Yeah, see, this is bad. Sometimes some things bacteria kind of help that, but more bacteria, if it gets in the wrong place, cause this. So these are decomposers, pathogens, ready to pounce when you get the chance. All right, so just to quickly review, bacterial metabolism, prokaryotic metabolism, that can be autotrophic or heterotrophic, and as self-feeders, they can be photoautotrophic or chemoautotrophic, depending on what they feed on. They can be, if they are heterotrophs and they live off of living organisms such as pigs, plants, humans, they may be gram-positive or gram-negative. In the case of human health, that helps us to diagnose and to prescribe medication. And they are classified by the three shapes: rod shape, circular, or spiral shaped: coccus, bacillus, and spirilla. Now, bacteria reproduce very easily. They just duplicate. We talked about it a little when we talked about mitosis, and that was binary fission. Remember that term? Binary fission. They simply duplicated their DNA and bloop, they split two in part. So bacteria can reproduce about every 20 minutes through binary fission. Every now and then, because they can reproduce so quickly, there is error. Now, typically the error is fixed, but about 1 in every 200 bacteria will have a mutation that sticks around. Okay, so mutations in bacteria can produce every 20 minutes. So 1 goes to 2, 2 goes to 4, 4 goes to 8. And in a day's time, even between brushing your teeth, you can have multiple replications, which is why you might leave the house at 7 in the morning having brushed your teeth fresh. And by the time you get to work, by 8 or 9, 10 o'clock, your breath stinks again. Those bacteria multiply. Okay. Yep. One good mutation for them may cause some bad news for you. The question we have to ask is, if they reproduce simply by binary fission with this mutation rate still, why haven't we been able to eradicate bacteria? Not that we would want to eradicate them. Again, they're just as much our friends as they are our enemies, and they compete against each other as well. Your bacteria typically evolve with you, and so they're friendly. But we know, because there is sickness, that every now and then bacteria are able to circumvent our immune system and overcome our healthy bacteria. So how do they do that? Well, they have to have a DNA sequence that allows them to make proteins that can counterattack our attack on them. Your body will attack it. That's why you get fever. That's why you feel the sore throat. Your body knows there's an infection, but it can't do anything about it. So one way genetic recombination occurs, and this is the whole power, right, of the bacteria, is that they can swap genes through something called plasmids. These are very important right here. We use them a lot in biomedical research. So what is a plasmid? It's a small supplemental circle of DNA inside. That's right, extra DNA. It's another chromosome, so to speak, but it's not. So let's see if I got a picture coming up here. So in conjugation, this would be your regular chromosome, that loop DNA that all bacteria have inside of them. Okay, that's your small DNA. A plasmid is this little tiny, it might have anywhere from 3 to 20 genes. This has 200 to 2,000 genes. 200 to 2,000 genes for life. This one might have 3 to 20. And the genes that it has typically are survival genes. So this plasmid can be very dangerous. It has information, inside information, on how to survive an antibiotic attack, on how to survive a macrophage attack by your body, all kinds of things. And the way it shares that is by direct transfer, called conjugation. It's what these two are doing right here. So let me show you a better picture. That's the DNA plasmid environment. If they release that, duplicate it, other bacteria can pick it up and adapt. But this is what I want to get to. So genetic recombination. So conjugation, this whole process is called conjugation. It's the top. Okay. So you have one bacterial cell, and notice you have the F, the Pro plus. Here's the other one. We call it the F minus because no male or female in bacteria. We call it the donor and the recipient, and the recipient generally is the negative, we call. So here we're Pro positive, here we're Pro negative, and they've duplicated that plasmid, and now one bacteria says, hey neighbor, you want some of this good stuff I got? And he shares a DNA sequence with it. Once they do that, this new bacteria is now a recombinant form of bacteria. So it has, F has the original genes. Now this one, which before had none of it, now has the plasmid with the Pro positive that it never had previously. Does that make sense? It's not too difficult, but sometimes I make it sound difficult. Here's one in action. Say, hey, check this out. Reach out and touch. So if you do not finish your course of medication, you take it for 2, 3, 4 days. I feel good. This is good stuff. I'm going to keep it for the next time I get sick. Guess what? You will get sick again. Why? Because you didn't finish the medication. You got to knock it out. There's a threshold level of infection, meaning the bacteria that you got sick with was probably already in you. Let's just say at a population of 2,000, and your body was able to withstand that. But once it hits something like 10,000, whoa, all of a sudden we need to do something about this. Your body mounts an attack, and the bacteria are growing and getting out of hand. You take an antibiotic, and you might knock it back down to 2,000, and you feel better. But those 2,000 are still there. And you know what? The ones you didn't kill, one of them will have a mutation. They'll multiply. They'll be able to share that mutation with the new others. Next time you take that medication, guess what? It's not going to work. Why? Because they've mutated and evolved. You did not kill them off. Finish the medication, kill off all the bacteria. Don't give them a chance to reproduce or mutate so that the next time they come back stronger. That's the important part of finishing the full course antibiotics that they give you for 7 days, 5 days, 10 days, whatever it is. Okay, transformation is another way bacteria can diversify. Why they haven't been wiped off the planet of the Earth through binary fission alone. And that's because there is DNA fragments from all over. You're chewing food, it's in your gut. Trust me, there's DNA on your hands right now. I worked in a biotech lab. I've worked in 2 biotech labs, and you always wear gloves. And the gloves are not for your protection. The gloves are so that you don't contaminate. When you work with DNA, you're working with small things, and you only need small amounts. But the small amounts you work with are about as much as the small amounts you might contaminate with. We have ways called PCR of amplifying the DNA to make billions of copies. And if you get any of your DNA or anything from your skin or from the tabletop in with that DNA you're trying to test, you're going to amplify your own DNA along with it. Especially in crime scene DNA, you definitely want to make sure your DNA does not get mixed up. So the goggles, the hairnet, the apron, and the gloves is not to keep you safe usually, although it does. It's usually to keep you from contaminating your samples. I'm just telling you guys, DNA is everywhere in small amounts. So the bacteria know this, and they're small enough to take it up. If the environment is good, all's peachy, nice temperatures, nice pH, this cell will go on probably replicating just like that. No need. But once you stress it, put an antibiotic in the environment, heat shock it, which we will do, or ice shock it, it freaks out the bacteria. And all of a sudden, he just starts looking for any kind of survival DNA. So the recipient cell takes up donor DNA. Now, we manipulate this in the lab, and this is truly what we do for, let's say, insulin production. We'll put genes in a petri dish and bacteria in a petri dish, and we will shock the bacteria. And when I say shock, I don't mean electric. Well, we can do it electrically as well. It's typically, though, with heat, extreme heat for about 30 seconds, and then extreme cold. And those extreme temperatures just cause the bacteria to go into a state of survival mode, and they take up any DNA from the environment they can. Now, if what you put in the environment is all DNA for making good insulin, then the bacteria will now have that gene for making insulin. It will incorporate that gene into its own genome, and you grow those. And we do this in the lab all the time, and the bacteria just multiply every 20 minutes. And as they multiply, they're also producing what? Insulin. And we can harvest that. And we have then human insulin that we can sell in a jar or bottle for injection quite cheaply. And who made it? Bacteria made it. And we did that through a process of transformation. We have transformed the bacteria by altering its genome in this way. This process is called transformation. So it happens in nature, it happens in your gut. Shingles, if you experience shingles, usually it's on your shoulders, but you can have it around your side as well. That's pretty tough. This happens when viral DNA gets inside of E. coli bacteria and transform the E. coli bacteria, and now they can cause shingles. Okay, well, we're at about 32 minutes. This is getting pretty long. I'm going to just kind of click through and let you... Hey, look, I think, yeah, we were looking at stomata. I believe it was your class, not the other. Here's the stomata underneath the plant. You can see, obviously, when it opens up to let CO2 and O2 in and out, that's an opportunity if there's bacteria in the environment for bacteria to get in and cause infection. Plants get sick too. This is my specialty, by the way. Disease-causing microbes of plants. All those. We have found some bacteria to help with pollution, actually, cleaning up oil spills and such. Tuberculosis, pneumonia, cholera, typhoid, gonorrhea, chlamydia, leprosy, plague, tooth decay, all these things caused by bacteria. Lyme disease. This is a really cool picture. This is a macrophage that was eating bacteria, but now it looks like the bacteria have multiplied to such a degree that it's overtaking the macrophage. Here's the brighter side of the story. Bacteria as beneficial and necessary. They are decomposers. They recycle nutrients. They provide nitrogen to plants in the nitrogen nodules. This is all underground. So each of these nodules here is filled with bacteria that are able to take in nitrogen. Plants cannot take nitrogen from the atmosphere. Bacteria can, though. And so they fix that nitrogen into chemical form so that the plants can get it. And the plants likewise give sugar to the bacteria. And so this is a mutualistic relationship here. Of course, in us, they help digestion. And cattle are able to digest. They have 4 stomachs, and the reason they can digest the grass, which we cannot digest, is because they have bacteria that allow them to break down the cellulose. They have cellulase enzyme. We do not have cellulase enzyme. So again, here's a nice adaptation for the different organisms. In our digestive system, they help us to produce vitamins K and B12. Of course, foods like yogurt. I mentioned the insulin already, through biotechnology and transformation of bacteria. So bacterial reproduction occurs through binary fission, but that's an asexual form. However, because of the mutation rate in replication and the ability of bacteria to share their DNA through conjugation or even take up DNA from the environment by transformation, bacteria can genetically modify or diversify over time. And this allows them, of course, to cause infection or to just simply co-evolve with plants and animals the way we see in these pictures here. That is, they depend on each other for their own livelihoods. All right. Oh, what happened to my picture? Can you guess who this was right here? I'll have to look up the boss. All right. Thank you for listening. 35 minutes is up.